All right, so we spent a long time talking about basics of psychopharmacology. The reason for that is I'm assuming most of you haven't taken a psychopharmacology course before. The last time I taught this, I don't remember any of you being in there and failing that class and having to take it again, right? So that's my assumption. But I think it's, like I said, I think it's important that we spend some time on this, think about how these things work. All of these principles are going to apply to what we think about after the first exam, right? After that sort of this foundational component, right? Once we get through this, we start talking about specific drugs, specific substances that you're going to use. We're going to think about these exact same principles, right? We're going to apply ligand receptor binding. We're going to think about agonists and antagonists. We're going to think about all those things, right? So today I'd like to sort of start with this concept of competitive antagonists, right? And this is really sort of important, and I think it's important for you to think about, at least here, right? How many of you are familiar with naloxone? Yeah, or some similar version of that, right? There's some other names of that. What do you use naloxone for? Anybody have an idea?
Stop overdoses.
Yeah, specifically though, overdoses of...
Opioids.
Yeah, it's got to be opioids, right? The reason it's opioids is because naloxone really binds in the same way that opioids bind to those receptors, right? They're shaped the same on some end, right? Some end of that's shaped the same, the end that plugs into that receptor, okay? The other sort of part of the molecule, though, is completely different, right? That part of the molecule doesn't give you that opioid experience. Brooke, instead, it gives you nothing, right? Which is great, right? Because if you've overdosed on opioids, like what's a thing that can happen to you if you overdose on opioids?
Stop breathing.
Yeah, that's the big one, right? Pretty much anything short of stopping breathing, you can come back from that. But once you stop breathing, then you're in some trouble, right? But if you can jab someone with some naloxone, that'll start competing because it's competitive. Has a nice high affinity for those opioid receptors. It'll start binding with those receptors, dislodging some of those opioids, right? And it gets bound, it stays bound, and it doesn't do anything. It doesn't do opioid things, right? So you're not going to end up with that overdose situation, okay? You don't have to raise your hand, but how many of you carry naloxone with you? I know a lot of folks carry some sort of, nobody does that. That's fine. I mean, some people do. There are a number of folks who, in this department, carry things with them. All right. Don't have to worry about that too much. Okay. Non-competitive antagonists, pretty similar, except non-competitive antagonists bind somewhere other than the receptor site. So those competitive sort of antagonists, they were fighting for the same location, right? And so when they're both fighting for the same location, only one of those makes it. The affinity of that molecule for that binding site matters. The concentration of that molecule matters in those cases, right? As to which one wins. Okay. So if you think about this, anybody a college basketball fan? Who are college basketball fans? One hand, two hands, great. So you have the sort of the NCAA tournament, right? There are some of those positions. There are like 60, like 8 teams now, but whatever, right? A number of those are what you might want to think of as sort of non-competitive spots, right? If you win your conference, you're getting one of those spots, right? The remaining spots that you call like at-large bids, right? You're competing sort of with all of those other teams that are still out there to try to get those spots, right? But if you win your conference, if Marshall wins the Mid-Atlantic Conference this year, right? They're automatically, yeah, I know, right now that's the face I make too. That's a no. Don't bet on that one. Accidents happen. That's how I'm assuming some of you got here. No, that was disparaging, but it's true, right? I mean, I'm not gonna like signal out which ones of you I think it is, but Emily, like in all honesty, right? Let's just be honest about it, right? The point is though, non-competitive antagonists are these guaranteed kind of things, right? Doesn't matter if that endogenous molecule is binding to that receptor or not, because this antagonist is binding somewhere else on the receptor. You can go through that door all you want. I'm going to go through this other one. There's another door just for me. I'm going right over there, and that's how things are going to work, right? So Montana, I could think of you, right, as like a non-competitive antagonist here, because you have this special door you can come through that no one else can.
This is right over there.
You're what?
My office is right over there.
I thought you said your house was right over there, and I thought that's
well, it doubles as that.
yeah. No, I understand that. So non-competitive, you have a different place you can bind. It doesn't. You're not competing for those same seeds, right? So that's what we have. If you take two drugs at the same time, guess what can happen? Well, sort of like three different things, right? They can either sort of be antagonistic toward one another, and they can kind of reduce the impact, right? So that's kind of like taking, what happens if you take some sleep medication and some caffeine at the same time, right? It doesn't sound like fun, but you can see how they might work in opposite directions, and you might just be just as well not to do anything. Sometimes you take two drugs, they have what are called additive effects, right? And they can add together. Now sometimes when you add these two effects together, you don't just get like a 1 + 2 situation, you get like a 1 + 2 + a bonus situation, right? They potentiate, okay? Right? So this is sort of like a, we can continue some basketball analogies, right? I mean, who was like a really great basketball player? Nobody knows. Somebody's going to throw out a name. Nobody.
Zion, he came back last night.
Who?
Zion.
Yeah, that's probably a bad example. Who knows what he's worth? I mean, in all honesty, right? Let's see what he does, right? Let's give him a chance. We can maybe pick another Pelican, right? Or a former Pelican. You're a Pelicans fan?
I'm a Duke fan, so I kind of just follow where they go.
Gotcha. Yeah, I guess. I thought if you're a Duke fan, you may not go with
Jason Tatum.
I forgot his last name. I'm looking at his face right now. Not your face. Nah, it doesn't matter. He's with the Lakers now. Let's use I don't want to use that classic example with the Lakers. Let's go back one more, right? Michael Jordan, great player. I think that's right. I mean, everybody's going to go with that. Scottie Pippen, also a great player. I think he's somewhat underrated. Just my take on Scottie Pippen for you there. The two of those guys together, I think they were probably better than either one of them, like independently, right? Which is true, but they were also better than what you would imagine like the two of them together should be, right? So you get like this bonus on top of that, which is really like what we call about potentiation, right? So you get some like extra sort of bonus. Okay. Some people you think might give you a bonus, they go to a team and they don't always do that. You don't always get as much bonus, right?
Well, no, because like Bryce Harper just got traded from the Nats.
Yeah, yeah. Bryce Harper's a great example.
And then he went to the Phillies and like got knocked out in like in a scrimmage game. Like they like threw the ball and he was out for a couple weeks and signed a $13 million deal. And they didn't even have a star pitcher.
Yeah, the Phillies were awful this year as well, and I don't know if you know what happened with the Nationals season.
Nationals won it, yeah. I'm a big Nats fan, so I was like upset to see him go because he was like the dude, and now he's not the dude anymore.
Yeah. Well, there you go. He was a great player when he was 16. No, he was, but that's about all he's done. Anyway, here you go. Antagonism, additive, potentiation. Not a big deal. We've talked about tolerance already. Okay. Diminished response as you keep taking the same drug. Why would you have cross tolerance? We gave you a couple of examples, right? Similar drugs, they activate the same enzymes, so you induce those same enzymes, right? These are just concepts we've already touched on. Acute tolerance, that's usually what we think about if you just take a drug once. Not a big deal. There are different kinds of tolerance. Sometimes it's hard to tease these apart, right? But they're like sort of different broad categories, which are like metabolic tolerance, right? So we're going to talk about enzymes. There's pharmacodynamic tolerance. This is when we're going to talk about up or down regulation of receptors. Okay, and it's sort of the third thing that we might talk about sometimes like a behavioral tolerance, right? And this is going to be related to like types of learning and so forth with your drug use. Classical conditioning plays a role. We'll talk a lot more about this as we go through. You can get, here's kind of a classic example. You give someone a drug, they've got a response, not a big deal, but the drug paraphernalia or the ritual is always associated with the drug, right? And you get that response. At some point, you can actually cut out the drug and you can still get some of the same responses just with those sort of sensory stimuli. Not a big deal. This is one of the reasons that this is just an example. For this case, they were looking at body temperature in different environments. This becomes a problem, right? If you always take a drug, either one you've been prescribed or one that you've not been prescribed, and you always take that in the same environment, and then for some reason you go to another environment and you take that drug, you can have different, more powerful effects, right? When you think about, like if I said, oh, somebody overdosed, you always think it's like in some random hotel room, right? It's like an environment they've never, it's like the, you know, right, it's an environment in which they've never been before. Maybe they were taking this drug for years, they were taking it somewhere else, and they had some different mechanisms that were kicked in gear if they were always doing this at home, and then they decide to, like, you know, take the show on the road, and they do that, and they can have a different sort of response to that, right? Their tolerance mechanisms might not be kicking in as much, and you can end up with some overdose situations. There's this whole thing about state-dependent learning. I always want to be careful about this, right? Because I don't want to recommend that you, like, right, Emily, you know where this is going, right? Like, let's say you came to class one day and you were not, let's just say you came to class one day under the influence of some substance, right? And you learned some amazing thing that I've said. And then I'm going to ask you about that. You're going to need to be under the influence of that same substance when you come back in for the exam, right? And so depending on what that substance is, I may or may not recommend that you do that. Does that make sense? Okay. Sensitization is important as well. Okay. Sometimes when you take a drug at the same dose repeatedly, you, instead of having tolerance, you become sensitized to this, right? And you actually have a more powerful response. Typically, when we think of sensitization, we think of that being a bad thing, right? On some level, you're thinking, well, I'm getting more for my money this time. The problem is, like, what are the side effects of that, right? Like, what if you take morphine or you take some opioid and you become sensitized to the effects of the, you know, depressed breathing? It's like a bad idea, right? You're like, I can take this dose, I can handle it, and then you're like, this time, well, I didn't breathe as well. So you don't want to do that. Pharmacogenetics, that's kind of a different thing. We've talked about this, making sure we look at your DNA to make sure everything is good to go. Kind of go through some of this. Some of these are things we've already covered. Just want to make sure this information is available to you. Okay. Any questions about that? I mean, that's exciting, right? So we finished that chapter. As I said, we're not going to talk about chapter 2. I know some of you are disappointed about that. Chapter 2 is very much a view of how your brain works, right? How many of you have actually read chapter 2? Maybe you've been in a lecture that has repeated the information of chapter 2. No. Some of you. Was it interesting?
I know what it looks like.
Yeah. Okay. What we do want to talk about, though, a little bit are neurotransmitters and hormones, right? Here's like the 2-second story. You've got neurons. They get excited. Electrical stuff goes through them. At the end, if they get excited enough, they're going to dump out some neurotransmitter. There's a whole lot of really interesting stuff that goes on there that you should learn about, but we'll just kind of leave it there, right? You think that's enough? Okay. So we'll talk about a few things here. I do want to introduce you to just a couple concepts. One's called the synapse. We'll talk about the synapse a lot, right? This is where one brain cell talks to another brain cell. And they're going to do this chemically, not electrically typically, so we're going to think about that chemical contact, right? Sort of the standard drawing is this this kind of bulb thing that comes down and contacts onto another cell, and then it dumps out little molecules of neurotransmitter, right, that bind to that other cell and cause it to get excited or to get depressed, right? It can be inhibited. It can be excitatory or inhibitory. Sort of depends on what's going on, right? Okay. So we call that sort of whole thing the synapse. Sometimes you'll hear things like the synaptic cleft. That's the space in between. Presynaptic cell is what's before the synapse. That's the guy releasing neurotransmitter. Postsynaptic is the guy receiving. Okay. You can make contacts on all kinds of places on those cells. Nothing to worry about. This is an electron micrograph. Use electron microscope. I don't know. Let's say this is somewhere around 15, 20,000 times magnification. This is that presynaptic cell coming down, and look at all those little, those are actually like balls filled with neurotransmitter. They're going to work their way down, dump them out here at that synaptic cleft. This guy, this dendritic spine, is going to receive those, and he's going to go, thank you so much for the dopamine. Guess what I'm going to do now? I'm going to tell 10,000 of my friends about dopamine and how you gave it to me, right? And so they're going to pass that signal through, right? These guys are constantly using ATP. Your brain is just like, I think about your brain, and if I were to like put like a gas gauge on it, like some parts of your body don't use as much energy as others, right? And you think like, but your brain is just constantly sucking energy, right? It's just constantly happening. Different synapses. We'll kind of breeze through some of this. Again, you can make synapses a lot of places. We give a fancy name to the synapse between a nerve cell, like a neuron or a nerve cell, and your muscle. We call that the neuromuscular junction. Why? Because it's the junction between the nervous and the muscular systems. I know we kind of made that fancy word there for you, but it's pretty straightforward what it means, right? Why can your nervous system cells talk to your muscles? They both speak the same language. They both speak that electrochemical language, right? How many of you have ever used like a TENS unit? Yeah. Yeah, right? And so they make you twitch, right? Because it's an electrical signal. Your muscles are electrically excitable tissues. Your nervous system is an electrically excitable tissue, right? Kind of the same things work. In fact, some of the same channels that are in your brain, some of those same ion channels are the, similar variants are ones that are in your muscles as well, right? And some of those can cause you real problems. There's like a whole sort of group of genetic disorders called channelopathies, right? And so that's when you have problems with your ion channels. The structure of your ion channel is not the way it should be. Sometimes that just causes you like a small problem. But when you start screwing around, particularly with the calcium channels, so you have like Ca2+, right? Carries 2+. Those are ones you don't really want to screw around with too much because they're involved in cardiac tissue. And so if your CAV channels, because they're voltage gated, so sometimes we'll call them CAV channels. If those CAV channels are screwed up, your heart probably doesn't work just right. If your heart doesn't work just right, anybody know what can happen? Yeah, you could die, right? It's not a trick question. You die. I mean, that's really all there is to it. Okay. You have 100 neurotransmitters. 100 chemicals have been identified as neurotransmitters. I'm not going to want you to understand all 100. I think a more reasonable number is like 97, 98 of those, right? There are 2 or 3 that just, it's not that important. In reality, probably, I mean, on the order of 5 to 10. 5 to 10 things is what you need to kind of think about here, right? I mean, they're not, if you start thinking about the big players, and they could be grouped, and we're going to skip over some of those groups, right? We're not going to spend a whole lot of time on like the individual sort of mechanisms of each of those, subcategories. I think that in and of itself would be an awesome class, right? Just a class on neurotransmitters, and we could just like sit for 2 hours a day and talk about like the structure of dopamine and related substances and how that activates dopamine receptors, D1 or D2 receptors, and all kinds of fun things. This class, unfortunately, only being a 14-weeker, for undergrads, we can't get quite as deep. I know, right, Emily? You're so disappointed. Maybe I'll teach you special topics, neurotransmitters, and other fun things.
There we go.
That's what we'll call it. Psychopharmacology Part 1 and 2. Make this a 4-semester course like a foreign language. I don't know. None of you. How many of you would rather take 4 of these courses than 4? They didn't even have to say. Natalie's like, already, I hate this class, but I really hated French. Oh, Spanish. Not everyone took Japanese. So, what are the big heavy players, right? These are covered in chapters, what is it, like 5 through 8, something like that in your book? Anybody know? Anybody have the book with them? Well, open it
They're
up.
not with me, it's in the car.
I wish this could catch my facial expressions. It just catches the audio and didn't quite capture my disappointment. Is it 4 through something? 5 through, you can look at the brief contents there.
5 through 8.
5 through 8. 5 through 8 is going to cover, so I actually was right. 5 through 8 is going to cover those specific neurotransmitters. What I would recommend that you do is look through them, because they're going to be interesting. I'm going to ask you about them. But in particular, when you have a presentation due, and let's say your presentation is on, I don't know, like psychomotor stimulants, right? And those psychomotor stimulants, you go, oh, this really messes with this neurotransmitter system. Go back and reference that chapter a little bit, right? And you can go back and you can take a look at that. So I would use those chapters, kind of briefly go over them, and right now, because I may ask you a few things about some of the main players on this first exam, but really use those as reference when you go through your presentations, right? When you're doing, who has LSD? Who has the chapter about LSD for their presentation? Let me rephrase that question. Nobody's hands are... I think that's the people who aren't here today. I think that's you, Natalie. Yeah, and so you know that works on the serotonergic system. So you're going to want to go back and look at that serotonin chapter, right? You're going to go back and go like, oh, okay, what's going on with serotonin? Let me learn a little about that and kind of bring that back to the forefront for you. That's how I would use those chapters. But the big things we are going to talk about, big things you've probably already heard of, are mostly going to be the guys on this chart, right? I mean, this is a pretty nice table. You have big categories. We think about amino acids, right? How many of you are familiar with the amino acids? There are a handful of those, right? You probably had to memorize those at some point. And all those like codons, right? I just remember AUG was methionine because that's what starts. Everything in August is the start of the school year. That was my... I know it was a great device, right? I forgot all the rest of them. Weren't that important. When we think about amino acids, there are really only two we're going to mostly focus on, right? And that's going to be glutamate, which is always going to be excitatory. Gamma aminobutyric acid, and yeah, I know that looks like a Y, but that's the Greek letter gamma. Okay, so don't call it Gamino, right? Try to pronounce it like a Y. Just call it GABA. That's how we abbreviate that. That's always going to be inhibitory. And then there's this guy glycine, and glycine's inhibitory as well. We don't think about glycine as much. It's not as big of a player on the inhibitory scene as GABA, but it's still something that comes up, and you kind of want to think about that. The next major group, and this is where we really, aside from acetylcholine, which doesn't seem to actually be on this list, which should. I'll have to add that.
It is right there.
Yeah,
Oh,
it's just
I was thinking,
circle.
oh, it's its own category. Okay, that's perfect. I was like, where is acetylcholine? I was looking for it indented. So the sort of big players, that we'll think about. Most of those are going to be called monoamines. Why are they called monoamines? It's based on the structure of that neurotransmitter, right? And right now I'm not going to ask you to think too much about the structure of the neurotransmitter, but all of your monoamines kind of start out as the same thing, and they go through maybe some similar steps of synthesis, and as you get along, there are branches, right? And depending on what enzymes are going on and what's happening and what that process is, you have a couple different steps, Brendan, and you end up either as, dopamine or norepinephrine, something like that. So we have the big guys here, dopamine. Most of you guys are familiar with dopamine. Norepinephrine. Epinephrine is the other sort of thing that's very similar to norepinephrine and epinephrine. They are often referred to as adrenaline and noradrenaline, right? It's kind of the same thing, so don't get too confused there. Serotonin is another big player that we'll talk a lot about. Serotonin's great. We've talked about SSRIs, for example. Low serotonin levels are typically associated with just not feeling good, right? You're going to be depressed, your mood's going to be down, you might be irritable, you might be aggressive, right? And those are kind of the serotonin stories in brief. We have histamine. How many of you have ever taken an antihistamine? Like almost everyone, right? I mean, Benadryl is pretty common. What happened when you took that? For most of you, there are a few weirdos probably.
Fell asleep.
Yeah, you fell asleep, right? So that tells you histamine is really important for keeping you awake. And so that's sort of an important neurotransmitter there. We get down to acetylcholine. Acetylcholine is involved in cortical activity, right? Keeping your cortex awake. So when you go to sleep, acetylcholine levels sort of drop in your cortex. Also, acetylcholine is used for muscle movements, right? If you get excited and you want to read about fun things that screw around with acetylcholine, you can read about like Botox and Black Widow spider venom, right? They both work on the acetylcholine system. They do different things with the acetylcholine system. I was going to say neither of them are good, but I guess it depends on how you use them, right? I mean, Botox has some medical and aesthetic applications that they use for wrinkles, migraines, TMJ. I think they sometimes use it for that, right? TMJ is used. I don't know that anybody's going out there and like using Black Widow spider venom medically, right? Like, hey, I don't think your arm is convulsing enough. Let me just put this thing in there, see what happens. One sort of blocks acetylcholine activity, the other overactivates it, right? So you have two responses. We will talk a little bit about ATP. Mostly we talk about ATP. We're talking about energy molecules there, and adenosine in general by itself. Adenosine is a sort of a byproduct of your brain doing its business, right? As adenosine builds up, you're going to get sleepy. You go to sleep, your brain's going to clean up all of that sort of metabolic waste. Guess what happens if you don't get enough sleep? I mean, I'm going to tell you, but if you're not getting enough sleep, you're going to get Alzheimer's and not remember. But you get Alzheimer's. I know, right? Yeah, that's the problem. They did this study with mice a few years ago, and they like kept them up, and all their brain trash didn't get cleared out, and they started developing not just symptoms of Alzheimer's, but the actual the plaques and the tangles and so forth. I know, right? So you guys should sleep the appropriate amount for you. May or may not be 8 hours. Okay. Some folks it's more, some folks it's less. You'll find out. Go to sleep for 8 hours, and if you wake up and you feel good, keep doing it. If you don't, adjust it one way or the other until you feel good. We're not going to talk about a lot of the neuropeptides. We will talk about some. We'll talk about some endorphins at some point. You guys have probably heard of that. We will talk a little bit about maybe orexin, BDNF. This sort of helps, brain function and structure and so forth. I don't think we're going to talk a lot about lipids. We might briefly. And then some gases. Primarily the gas we'll think about is nitric oxide. We won't think about those other guys too much. Nitric oxide is a great signaling molecule, right? One cell does something, it wants another cell to know it did it. It can shoot out some nitric oxide. We'll get back to it. Okay? Nitric oxide is lipid soluble. Also a vasodilator, right? So you kind of have that in a variety of substances. Emily, I'm sure you can tell us about all the people who come in and get substances who work on nitric oxide. I mean, I mean, how do you figure out if something's a neurotransmitter, right? Well, basically, it's a chemical. The cell makes it, releases it. It gets inactivated somehow, and the causes some activity on the other cell. Not a big deal. Vesicles, synaptic vesicles can hold a number of neurotransmitters at one given time. Each cell can release multiple types of neurotransmitters. This is important, right? Previously, folks had always thought like that cell only releases acetylcholine. Well, no. That cell can release acetylcholine and a handful of other neurotransmitters as well, right? So your neurons are quite flexible in that, which becomes a little bit of a problem, right? Anybody know where you take most? I mean, like, where do you put most psychotropic medications when you take it? In your mouth. That's the answer, right? But most of them come in pill form, right? So you get a bottle, you take it home, you take it. Where does that substance go then? Well, it goes everywhere, right? And then it gets in your brain. And a great example of this is did we talk about Parkinson's disease and schizophrenia in this class, right? So that's a great example of this, right? When you're trying to treat someone's dopamine levels, and their dopamine levels are too low in one brain region, so you give them a substance to increase dopamine levels. It sounds great in that area that's too low. The problem is, in the areas that were normal, we now are higher than we need to be, right? And so we can cause some problems. So when you take most psychotropic medications, most medications in general, they're administered systemically, right? And they don't have these sort of targeted things. One of the coolest thing that's going on, and maybe someone, Montana, when we get to the methods chapter next week, if you're interested in talking about bespoke receptors for a few moments, if you want to do that, you're welcome to do so. It's some of the coolest stuff going on right now. I mean, it really is. I'm excited for it because I'm just waiting to. I don't know. My head is going to be like an Xbox controller. I'm going to have buttons all over it. I'm going to turn on this brain region now. Let me activate those. You just take pills, right? It's going to be exciting. It's cool stuff. All right, remember that thing I drew you that looks just like this, right? I told you this is exactly how they draw, which is actually not an inaccurate representation. The problem with this is they don't draw like all of the other stuff that's jammed around in there, right? And you kind of have this idea that there's like this empty space in your brain. I mean, in some sense there is, but in some sense there's not. It's kind of exciting. Most neurotransmitters are made like their final steps are done down at the terminals, but we have to do some of the work here in the cell body. We can send things down and then go through that final sort of step, right? So it's like, I don't know, if you buy, how many of you buy furniture from IKEA, right? They sort of ship you the parts, right? And then you have to do the final assembly at your house. They did a lot of the stuff somewhere else, shipped it to you, and then you do the final assembly, right? And so that's how it works with neurotransmitters, right? That final assembly is typically done down at the terminals, but we got to send the stuff down there. We'll send stuff back too. We'll recycle things. This is an expensive process, and we want to save as much of it as we can, right? It's like reclaimed barnwood. Neuromodulators are sort of interesting. They don't in and of themselves cause activity, but once the activity is going, if there's a neuromodulator, it can cause some change of that activity, right? It can either enhance, prolong, reduce, something like that. Okay. This distinction between a neurotransmitter and a neuromodulator is kind of fuzzy sometimes, so I don't really want you to spend a whole lot of time like trying to figure out which substances go in which category. Think about this. If it by itself can do it, it's a neurotransmitter. If it needs a buddy, it's a neuromodulator. Calcium. I'm not going to hit you too hard with this, but once you get down to the end of this neurotransmitter, there's got to be an electrical signal to cause these vesicles to undock, float down to the synapse, and then dump themselves out. So that's cool, right? Vesicle recycling, we talked about this. Why is this important to you? There are certain drugs you're going to take that are going to screw around with your ability to store things in your vesicles, to create vesicles, to pull things back into the cell that were dumped out by vesicles, to create the neurotransmitter itself, right? One of the big treatments for Parkinson's disease, again, is L-DOPA. That's a precursor to dopamine, right? So it's kind of like, we want you to build houses. So what do? We give you 2x4s, right? Okay, so we give you the precursor to that, and then you have to put it into a house. I'm not going to ask you too much about synaptobrevin and so forth. Oh, well, here's botulism. That's a fun one, right? We could spend a lot of time talking about, like, clathrin-coated vesicles and so forth. I don't think that's going to be very helpful for you. If you're interested in it, this is fascinating, right? So these vesicles, to kind of pull them back in through endocytosis, to suck stuff back in, there's a special set of proteins and so forth that need to be involved in that, right? And that can get pretty complicated, but it's exciting. But I'm not going to ask you about it. Ultra fast, some of this stuff happens more quickly than others. That's what I want you to take away from that. And then sometimes we think about things that are what we'd call a kiss and run situation, right? So this vesicle will kind of go in, briefly dump, and not completely merge, just kind of, right, touch and release and go on. There's bulk endocytosis. I'm not going to ask you too much about that. We talked about lipid solubility, right? So you know about that. Retrograde messengers. These are actually kind of interesting. Typically, these are gases or lipids. Why would they be gases or lipids? They don't need a special door to get through the cell membrane, right? And so going in reverse in the other direction, you don't really have to do anything to prep that, right? It's just going to slide right in there. And again, typically nitric oxide is going to be your guy there. Okay, so you can see here's that retrograde messenger. When that postsynaptic cell gets activated, sees something going on, it can send information back to that presynaptic cell in the form, again, typically of nitric oxide. Are nitric oxide synthase, are they still big? Like I know a lot of people use those in like weightlifting supplements. Anybody know? That's still, a lot of people still use those, right?
It's like in pre-workout.
Yeah, yeah. So you get the yeah. I know, right? Whatever that is,
Yeah.
Put that in quotes. I mean, again, nitric oxide is typically thought of as a vasodilator. That's why.
It pumps up other stuff too, though.
Yeah, yeah, it does. So if there's something else that you might need at some point in your life or someone else's life to work at a different capacity than it currently is, nitric oxide boosters can help with that, right? Like Viagra, for example, is kind of works through that nitric oxide system, right? 'Cause it's a vasodilator. This reminds me, none of you have probably been to the Tri-State. Have you been to the Tri-State Psychology Conference?
No.
So we've had it here a couple of years, and they usually have me host the Psychology Jeopardy section, right? And so I get up there and I ask questions and teams buzz in and they answer. I'm like, whatever, right? And I try to do my best Alex Trebek impersonation, right? And so a couple of years ago, I guess it hasn't been on our, they rotate where it is, like between us and some other schools in West Virginia. Typically, there are a couple of Ohio schools. But there was a question and the answer was erectile dysfunction. We had like 5 teams of 5 players and nobody buzzed in. And I was like, yeah, nobody wants to admit to knowing about that. Which most of the professors who were there thought it was hilarious and the students didn't laugh. I thought it was a great joke. But no one, everybody was kind of just sitting around. Nobody was answering that one. It's the only question where time ran out and nobody had an answer. I guess you don't want to get too excited about that, right? Like really buzz in fast, yeah. Erectile dysfunction. Autoreceptors, we should talk about these a little bit. Basically, An autoreceptor's job is to make sure that you've released a neurotransmitter, right? So this is sort of like a, you've sent out dopamine. You want to make sure you've sent out dopamine. So you might have on the cell that released dopamine an autoreceptor, and it can detect dopamine. Once you've dumped enough dopamine that it's working its way back to that autoreceptor, you know, Brooke, I've flooded that synaptic cleft with dopamine. I can stop. There's enough dopamine out there that I'm seeing it come back to me, right? And so that's kind of like, that's some good information there. There are different kinds of autoreceptors based on where they're located. Don't worry too much about that. They're really doing the same thing. Yeah, they're just working on that system. This is a little nuanced. I'm not going to ask you too much about heteroceptors. All right. We should think about the inactivation of a neurotransmitter, right? And so we've talked about this some. Let's talk about it a little bit more. Typically, we're going to do a few things. We can do a process of enzymatic degradation of the neurotransmitter. We've already talked about enzymatic degradation before. We've thought about that mostly in the liver and breaking down drugs, right? We're thinking about something a little different here, different location, same concept in some sense, right? Or we'll think about reuptake, okay? So let's think about reuptake for a few moments. This is when you've released a neurotransmitter and you've decided to reach out and grab that up and scoop it back into the cell that released it, right? Okay. Most of you don't really think about reuptake, except when you think about SSRIs, right? What's the R stand for? Reginald? No, it's reuptake, right? And these are reuptake inhibitors, selectively for serotonin. That's in the wrong order, but the right letters. So selective serotonin reuptake inhibitors, right? So these guys block that serotonin transporter. We're not pulling serotonin back into the cell. Guess what serotonin's doing? More of what it normally does for a longer period of time, right? It's just hanging out there doing its business. We're blocking that inactivation. We're blocking that where we stopped, right? So it's kind of like, how many of you are soccer fans? Right? Yeah, so like what happens at the end of a soccer game? The ref sort of inhibits the end of the game, right, by adding injury time. And so, the game's going to go for longer. What are those soccer players doing? Same stuff they were doing the other 90 minutes. They're just doing it for more minutes, right? And so that's kind of like an SSRI. Serotonin's doing its same business. It's just doing it longer because we've blocked the whistle at the end of the game to call everybody back into the field house, right? I almost said dugout, and then I thought that's the wrong sport. All right, here's a great example. No worries. We will think about drugs that do block transporters at some point, right? We've talked primarily about those serotonin, but there are also SNRIs, right? And those work on the norepinephrine system. Basically do the same thing that serotonin reuptake inhibitors do, but a different neurotransmitter, right? Blocking reuptake isn't always going to be the best approach. The reason for that is you're not you don't know, things aren't always stopped by reuptake, right? Sometimes they're stopped by other things. And so like acetylcholine esterase, for example, this is an enzyme that breaks down acetylcholine. If you wanted to increase acetylcholine, you might inhibit the activity of that enzyme, right? So you might do that. We talked about MAOIs. Did we talk about MAOIs in this class? So. You guys couldn't go to wine and cheese parties? Okay. Yeah, that's a monoamine oxidase inhibitor. That's inhibiting the enzyme that breaks down monoamines. Who remembers table 3.1? I just showed you that a few slides ago. That's the one that had that chunk of monoamines on there, right? Like dopamine, serotonin, right? So the problem with a monoamine oxidase inhibitor is it sort of prevents the breakdown of all monoamines. And again, it knocks you out of getting wine and cheese. What do we want to think about receptors? A couple different kinds of receptors. Some of those are direct, right? That's what we call an ionotropic receptor. Something's going to bind, open an ion channel, stuff's going to happen. Metabotropic are a little slower because you have to send something somewhere else, right? You have to have kind of a second messenger system there. If you guys aren't too familiar with this, I'd spend a little time thinking about it, particularly when it comes to your drug, right? And so there are receptors that are activated by different drugs, right? Sometimes we name the receptors that, right? So there are muscarinic and nicotinic receptors, that are activated by those drugs, at least in the lab. Doesn't mean that they're allowing nicotine into the cells, it just means that's activating them. Just don't have to worry about too much of that right now. When ion channels open, when those receptors activate those ion channels, whether it's directly with ionotropic or it's through that cascade with metabotropic, we're going to allow ions to flow in and out of the cell, depending on which door is opened. These allosteric sites, they're sort of important. These are places where modulators can bind. We talked a little bit about modulators before, right? And they can bind and change the activity of the cell. So you can get many responses out of the same receptor, right? By using different modulators. That's the point here. It gives you some flexibility, right? So even though you maybe only have a handful of receptors, you can get sort of different activity out of them depending on the presence of a negative or a positive modulator. Okay? I know this is kind of vague. We're not really giving you a lot of examples here. But when they come up in the later two-thirds of this class, you'll kind of know what's going on. I hope some of you get really excited about protein kinases. Not sure who that's going to be just yet. Somebody, right? Kaylee's already. She's like, I have that tattooed across your shoulders. Protein kinases. It would be more exciting if you had them like tattooed at your like you know, and then you could like, it'd be pretty cool, right? If you had like two different, and then you could like bring them together, right? And you see, you're thinking, you've got this. And then like on your elbow you could like tattoo the enzyme name, right? And you just like, here's the activity. No one else thinks that's as cool as we do. I don't know why. Nobody's signing up for that, right? I opened my own tattoo shop. You don't get to choose what it is. I'm going to give you a cool science tattoo. Come on in. Half price if you let me pick what it is. So without really bogging you down with a bunch of like biochemistry, right? Because I don't really want to do that. But at the same time, I want you to think a little bit about it. But like just enough so you think you understand it, but you probably don't. But just enough so you think you do, so that later when you're reading about something, you're like, oh, I think I have an idea of what that is, right? Basically, there are a lot of steps involved in these second messenger systems, right? Neurotransmitter binds to that receptor. All right, fine. It sends like a G protein somewhere else, works on some other enzyme, sends out a bunch of second messengers in this cascade, activates protein kinase. There are all kinds of different protein kinases, right? There's like calcium calmodulin kinase 2, right? That's a fancy name for a very specific kinase, for the other specific names. That's going to take some protein, start punching around on that, changing its shape, making it do different things, and then stuff's going to happen in your cell. Okay? They're not going to hire me in the biochem department to teach that, are they? But I think that was the Cliff Notes version. Okay? You're not going to answer an essay question about second messenger systems with that information, but you're not going to be completely lost when you read about something, right? And when you read about a drug and it says, oh, it messes around with this protein kinase, you have some idea of where that is in this cascade, right? Here are some second messengers. This is probably the one you hear about the most, right? Okay. Cyclic adenosine monophosphate. We talked about adenosine before. We talked about the triphosphate version. Guess how many phosphates that had on it? 3. Guess how many the monophosphate has on it? Yeah. Then there's a diphosphate too. How many are on that? I know, right? We're so much here is working in your way. And it works on this PKA, right? This protein kinase A. Not a big deal. Okay. Here are some other second messengers and protein kinases. At this point, I'm not going to expect you to memorize specific protein kinases, second messengers, and so forth. I want you to be aware of the concept of second messengers and protein kinases. Once we get into specific systems, I may say, let's think about this specific kinase, right? And then we can think about that specific activity. And you might want to come back and reference this information at that point. Okay. When's the exam? The week after next?
Mm-hmm.
Is that accurate?
I don't know. I hope not.
It is, right? No, I hope so. Because next week we want to do all methods, pretty much all next week, and then you'll be ready. Is the exam Tuesday or Thursday? It's Thursday, right? And that gives me Tuesday to talk about addiction. Seems reasonable. So I've got 3 class periods left after the 13 minutes I have left now. That's not so bad. We're right on target. Do we want to think about neurotrophic factors? At some point, you will need to think about neurotrophic factors. We already talked about BDNF today a little bit. These are things that are important not just during development, but maintenance of normal brain activity, right? If you screw around with BDNF, that's a bad idea. Up or down, right? Okay, supposed to have a certain level of it, keep it there. Let's see. We talked about precursors. This is actually pretty nice. This is a nice diagram that sort of shows you the different ways that drugs can change how neurotransmitters are created, stored, released, inactivated. There's a lot of information on here. We've talked about most of this. What I would do is I would keep this slide like in mind, because when you go and you do your presentation on your drug and you find out, oh, it inhibits the release of a neurotransmitter. Okay, well, let's think about how that works. Or it actually, inhibits storage in vesicles, or it blocks receptors, or in fact it inhibits degradation, or blocks autoreceptors, for example. You can think about what effect that has on the neurotransmitter action. Okay, so that's actually a really good slide. Here are some very specific examples. I don't want to necessarily go through these with you, all right, but here's some that can inhibit enzymes, block enzymes, block transporters and reuptake, storage vesicles, to release neurotransmitter, stimulate that release, right? Mess around with autoreceptors. Okay. The end result is that these drugs can kind of be lumped into 2 broad categories. We talked about agonists. We talked about antagonists. Right. We're just kind of reinforcing that concept here. What do you want to know about synaptic plasticity? This is important. We've already talked about synaptic plasticity, right? We talked about it in terms of tolerance and upregulation and downregulation. Okay, that's really all that's going on here. We're changing the structure of that synapse in terms of the proteins that are available, right? What receptors and what ion channels are available at that synapse. Okay, why is that going to change? It's going to change because the environment changes. Drugs you're taking are changing. Internally, the chemicals you're creating are changing, right? Learning and memory is a great example of synaptic plasticity, right? Okay. Not a big deal. There are a variety of kinases that are involved in this, right? I'm not going to ask you to know too much about that. Spines grow and change. Spines get clipped off like dendritic spines. And there's like a pruning process. So the shape and structure of your brain cells change over time. I mean, they can change quite rapidly as they need to, right? Don't worry too much about this. A whole bunch of stuff about synaptic pruning. We should briefly talk about hormones. Hormones work just a little bit different, but they're still kind of the same idea. The difference with hormones is they really need to get like into the cells to cause changes. Neurotransmitters kind of work on the surface. Hormones really need to get into the cells so they can change like DNA production and stuff, or protein production, reading that DNA. Think about this with the endocrine system. Here are some places where hormones are created, things we talk about when we think about the endocrine system. Adrenal glands, those are sitting on top of your kidneys. Not too big of a deal. How many of you have some glucocorticoids floating around right now? Anybody stressed? Let's make the exam Tuesday. What happened to glucocorticoid levels is skyrocketed, right? Or for some of you, they may have dropped because you're like, I'm out of this class now. I don't have to worry about it. I'm done. There are your glucocorticoids. Typically, we'll talk about cortisol. Corticosterone is in, like rodents. What's the difference? I'm sure there's some small differences that are important to some people. They're not going to be really that important to you, right? Because they're doing functionally the same thing. Okay. Why are they called steroid hormones? They're made from cholesterol. It's just a name they get. Later, we are going to talk about anabolic steroids, right? Those are different in some sense. Okay, and so we want to keep that in mind. So if you're like taking a steroid, right, like prednisone is different than like the juice or the cream, right? The cream and the clear. Nobody's apparently like a Barry Bonds fan. There's this great documentary. Oh, man. What's the name of it? It's about, you remember the doctor, and I'm using air quotes here, who was selling like A-Rod, the steroids in Miami, and he got busted, and him and his dad got busted. It's like a big, Tony Bosch, I think was his name. Anyway, there's a funny documentary on Netflix about that. You wouldn't think this would be funny. The reason it's funny is they've got like a bunch of 10 and 12-year-olds acting out all of the scenes in the documentary. And so they've got, and they're all like, when they're interviewing people and they're like having the adults say the words, and then the kids are just like mouthing the words toward each other. And it's hilarious. Like there are a couple of guys who have like arm tattoos, and so there are these like 10-year-old kids with like bald caps and like fake arm tattoos running a tanning salon. It's just that was a good use of my time. That was if I could remember it was just a great story. It was a crazy story. Steroids made me think of that. Other sort of steroids, right? Coming out of your gonads, that's where a bunch of these come from, steroid hormones. You got your estrogens, progestins, and androgens. You've got that thing called the pancreas too. If you've got diabetes, right, you're probably aware of your pancreas. If you don't have diabetes, you're probably not aware of your pancreas. I mean, that's usually right. I mean, if you don't have diabetes, why should you worry about your pancreas? You probably shouldn't. Unless you think you're going to get diabetes, then you probably worry about it. Otherwise, it's just sitting there doing its business. Your thyroid gland, pineal gland, pituitary gland. That's kind of an important one, actually, up in the brain. We won't talk a lot about these, but it's nice to be aware of them. Let's see. Other things we should think about. Should we think about oxytocin? Probably not today. Maybe at some point you should think about oxytocin. It's a fun story. I love telling the story, but it probably doesn't work as well here. Now here's a great example. Why are we thinking about this? Melatonin, for example, can have some psychoactive properties. Melatonin is a hormone. That's kind of interesting. There are some interactions with hormones and drugs that you would take, so you might think about those as you're thinking about the your presentations that you put together later, as we talk about specific drugs and how that affects what's going on as your hormone levels change. There is one group of the population that seems to have higher hormone fluctuations in a short period of time than other groups, right? Okay, so women between puberty and menopause are typically going through some pretty rapid hormonal changes, like over the course of a 28-day span. Doesn't seem to really be for those pre-pubertal individuals. Post-menopause, women don't really see that as much, and men don't see those sort of like week-to-week fluctuations, right? Here's sort of a great example of those various hormones and how they're fluctuating throughout that menstrual cycle. Okay. And taking cocaine at different phases of this menstrual cycle can have different effects, right? So there you go. That's interesting, isn't it? You think about, when do I want to take cocaine so I get the most bang for my buck, right? If you're having menstrual cycles. If you're not, you take it whenever you want. It's going to be the same effect, right? I mean, if you got a choice. Don't worry. And this is sort of the same idea with. I mean, to be honest with you, the good old estrogens are pretty helpful, right? I mean, they really are, Kaylee. They'll keep you from getting schizophrenia. I know, right? That's probably one of the most amazing things about estrogen. Most of the time you think about schizophrenia happening like, in your mid-20s, right? Once you get past that point, you're like smooth sailing. And that's true, except for there's this small blip in females, like after menopause, where there's an increase in the diagnosis of schizophrenia, and that's because your estrogen levels are dropping then. I don't know if you found that interesting. I did. All right, any questions about that? That's the actual end of that chapter. We made it through chapter 2. We made it through chapter 3. No, we made it through chapter 1 and chapter 3. And